All right, we're going to continue on with our hemostasis story here. And so as I mentioned on the last video clip, hemostasis involves three major phases. One is vascular spasm. The second one is formation of a platelet plug. And platelet plugs sound pretty good, but those are really just temporary seals in a blood vessel wall that has been ripped open. The third phase is coagulation. That's the formation of an actual blood clot that more permanently closes off tears in blood vessel walls and prevents blood loss long enough to allow the blood vessel wall to be regenerated, to be repaired. This is also the more complex of the three phases that we'll be talking about now. But try not to get overly lost in the details. The diagrams in your textbook and so forth are going to look kind of scary, but it's not quite as scary once we start working through the story a little bit. All right, so when we form a blood clotting is also known as blood coagulation, and it helps reinforce your platelet plug with fibrin threads. Fibrin is a protein. So these are thread-like proteins. So as you guys know, there are many different types of proteins in the body that have lots of different roles. We've been talking about hemoglobin as a type of protein that transports oxygen around in the body. And here's fibrin, a completely different type of protein that's involved in coagulation. So essentially a blood clot converts your blood in that area from a liquid to a gel. You guys know what clotted blood looks like and how it's a lot thicker than normal blood. All right, so it's actually a pretty complex multi-step process that leads to blood coagulation. And as you might imagine, this process has to be very carefully controlled. You don't want to trigger this. You want to trigger it easily enough that you can seal off tears in blood vessel walls and prevent you from bleeding to death. But you don't want it being triggered so easily that you just have random blood coagulation events happening in your body at any given time. Because as you know, a blood clot that randomly forms in the blood vessel can be bad news for you. Can actually kill you if it's in the wrong part of the body. All right, so as we're going to see, coagulation involves a multi-stepped chain reaction or cascade that takes place, and it involves a number of clotting factors. Most of these are proteins. Not all of them, but most of them are proteins made in the liver. So liver function is very important for blood clotting. You may have heard of that before. All right, so in the blood, circulating all of the time, you have these clotting factors, but they're in the form of procoagulants. So pro means before they're coagulants. So they're actually, come on stylus, they're inactive at this point, but they're poised and ready to go, as we'll see. Very quickly they can be converted into coagulants that will lead to the formation of a blood clot, like you see over here, where you have this fibrin protein meshwork there you see in the light pink, and it has trapped red blood cells, and then your platelets there have been colorized in yellow. All right, so there are 13 different clotting factors, and they're designated by Roman numerals 1 through 13. And no, they are not named in order of the particular step that they occur in the process. They're numbered according to when they were discovered, which makes things a little bit confusing. And then finally, you need vitamin K. Vitamins, again, are organic molecules that don't fit into the amino acid, sugar, lipid type categories. They're organic molecules that you must take in from your diet because our cells cannot manufacture them, but they need them for various processes. And vitamin K happens to be an organic molecule that is needed in order for the liver to be able to make 4 different clotting factors. So you may have heard of that before. You need vitamin K for proper blood clotting capability. Okay, There are three phases of coagulation. Now don't get confused with the three major stages of hemostasis in general. Here we're talking about the third phase, coagulation, and that third phase is itself divided up into three major phases. One of those involves forming an enzyme called prothrombin activator. This is an enzyme, and it is present circulating in your blood in a inactive type form, or it has to be assembled as part of the coagulation process. It gets formed through two different pathways that we'll be taking a look at. One's called the intrinsic pathway and the other one's the extrinsic pathway. Once you make prothrombin activator, that's step number one. Prothrombin activator is an enzyme, and it takes a procoagulant called prothrombin and converts it into the enzyme thrombin. So prothrombin, we said when we see the pro prefix, that means it's an inactive form. It's not doing anything. It has to get converted into the active form thrombin. And then thrombin is an enzyme. You know, enzymes do chemical reactions. They build things, they break things down. Thrombin catalyzes. Enzymes catalyze chemical reactions. They speed them up. They make them more likely to happen. It converts fibrinogen. So fibrinogen is an inactive clotting factor to fibrin. That's the active form that forms the mesh of a clot. So this is what we're trying to get down to here. We're trying to get to these fibrin threads, but it's a multi-step process to make that happen. And again, the reason why it has to be so complex is you don't want to just randomly form blood clots at any given time. All right, so on this last slide over here, we said there's an intrinsic pathway and an extrinsic pathway that leads to the formation of prothrombin activator, which is an enzyme. Oops. All right, and so this fairly complex looking diagram over here is showing that to you. Now keep in mind, we've already done this, vascular spasm. We've already made a platelet plug, and the platelets in the platelet plug start attracting these clotting factors to cause coagulation or blood clotting, this third phase, to take place. So this is where we are now. There are two different pathways that are known for triggering the formation of a blood clot. One of those is called the intrinsic pathway. All right, and as you can see here, this gets triggered by a blood vessel rupturing. The endothelium, the inner lining, gets ripped open, and that exposes underlying tissues. So, you know, blood vessel walls are at a microscopic level. They have multiple layers, and one of those layers includes connective tissues where you have collagen. So exposed collagen attracts platelets, like we've heard about before. And then the platelets actually start attracting some clotting factors. All right, so one of the things those platelets will attract is this one. That's the Roman numeral 12. This is factor, usually these are called factor 12, or the next one is factor 11, XI. If you follow these arrows, factor 12, this little arrow over here, gets converted to 12 with a little A subscripted there, and that's the active form. That's what that stands for. This is active or activated factor 12. Okay, and then notice if we kind of follow the arrows here, once factor 12 has been activated, it functions as an enzyme, and it's going to take factor 11, which is in an inactive form. It's in a procoagulant form, and it's always circulating around in your blood in that form, not doing anything. But when you have activated factor 12, that clotting factor takes factor 11 as a procoagulant and converts it to active factor 11. All right, and then once that has formed, once you have activated factor 11, that functions as an enzyme, and it takes factor 9, which is always circulating around in your blood in an inactive form, and combines it with calcium ions that you see over here, and also uses another activated factor, factor 7, that you see over there, brings all of those things together, and that helps form activated factor 9. Okay, so let me erase this because it's getting kind of messy, so you might need to pause if you're trying to write anything down before I erase it. Okay, so you can kind of see where this is going. One, you have all these inactive clotting factors in your blood all the time, and one gets activated and then it functions as an enzyme that activates the next type of clotting factor. And then that gets activated. Once it's activated, it functions as an enzyme that activates the next clotting factor. So you kind of more or less have a cascade or a chain reaction that's taking place here. All right, if we keep following the story on this one called the intrinsic pathway. The reason why this is called the intrinsic pathway is because all of these different factors that are involved in this pathway are natural normal components of the blood, or they are intrinsic to the blood. That's where that name comes from. All right, so kind of continuing on this side, once you have activated factor 9, your platelets that form that platelet plug, they release a chemical called platelet factor 3. That plus your activated factor 9 takes factor 8, another procoagulant in the blood, converts it to activated factor 8. Okay. Now, activated factor 9 and activated factor 8, they form a complex, and they together function as an enzyme, if we keep following our story over here, which takes factor 10 that's in the blood and activates it and forms activated factor 10. Okay, so again, just to kind of review, intrinsic pathway, we've gone through this multi-stepped process, and the goal here was to get to this activated factor X, factor 10, I'm sorry, down here that you see. It just took a multi-step process to make that happen. Now, if you're following along this pathway, it's a multi-step process, so if this one up here never happens, then neither will these other ones in this pathway. You've got to activate factor 12 up here at the top to make those other steps happen. And also, this is a positive feedback type thing, because as I said, these activated clotting factors are actually enzymes. Enzymes can go around and do a chemical reaction, and then they do another one, and then they do another one, and then they do another one. They can be reused. So like factor 12, once you have activated factor 12, all of your activated factor 12 molecules can go on the hunt for factor 11s and convert a bunch of those over to factor 11s that are now activated. So now you have more activated factor 11s than you did factor 12s. So they can find factor 9s and activate a whole bunch of those. So you're increasing your number of different types of these activated clotting factors as you move down the pathway. All right, let's look over here at the extrinsic pathway. It's actually quite a bit simpler. Let me erase all of that. The extrinsic pathway is called that because not all of the factors that are involved in this pathway are actually located in the blood. They're not intrinsic to the blood. So when you have a tissue, so when you rip open blood vessels, you're almost always destroying or injuring surrounding tissues, and cells in your tissues, when they get injured, they release a protein called tissue factor, and tissue factor helps set off this extrinsic pathway. Notice calcium is involved. You need calcium for both the intrinsic and extrinsic pathway, so calcium can also be considered to be one of these clotting factors as well. Tissue factor, when it's released from injured tissue cells, if you have calcium available, it triggers factor 7 to become activated factor 7. Okay, so this basically functions as an enzyme that activates factor 7. Okay, and then that forms a tissue factor activated factor 7 complex. They form a complex with each other. They're able to get together. And then that complex is able to take factor 10 and convert it to activated factor 10. So you see, this is accomplishing, this over here has accomplished this, just like all of these multiple steps did over on the intrinsic side. It's just over the extrinsic pathway is a little bit less complicated. All right, so you've got these two different pathways that lead to factor 10 becoming activated. Now we said, let's back up over here for a minute. We said our 3 phases of coagulation. The first one, we need to make prothrombin activator. So we're almost there. Once you have this activated factor 10, it functions as an enzyme, okay, and it takes factor 5 and converts it into activated factor 5. It activates it. And then this calcium ions that are present in your blood and body fluids, and this good old PF3 up here that your platelets made, all of these things assemble together and they form prothrombin activator. So it's a complex of these 4 different things that get together and make prothrombin activator. Okay, so we said that was where we were trying to get to. That was our first major step of coagulation. All right, don't freak out over all of these different clotting factors and the order in which they're used. I do want you to understand what sets off or activates the intrinsic pathway versus the extrinsic pathway. And in general, I want you to understand that clotting factors exist in inactive forms in your body fluids, and they become activated by the preceding factor in the pathway. So one gets activated, it activates the next. That activated factor then activates the next. So you have these multi-step processes. I do want you to know that you have calcium ions that play an important role as well. And incidentally, when we're saying that these clotting factors exist as procoagulants, kind of in a nutshell, what that means is they have 2 parts. So here's your procoagulant, and when you have an active clotting factor that becomes an enzyme, it comes along and clips a portion of that procoagulant off. So this goes away, and then what you have left over here, that is now your active clotting factor. So it's essentially like it clips off a portion of it, and that's what activates it. All right, let's continue on then. So once you have prothrombin activator enzymes around, now you're able to finish up this process of making a blood clot. All right, I think this slide here just has some written information summarizing what I already talked about. Your extrinsic pathway, as you can probably tell from the diagram, is going to be faster because you don't have as many steps. And typically also the intrinsic pathway and the extrinsic pathways happen together, because the intrinsic pathway is going to happen anytime you rip open a blood vessel and expose the collagen in the walls of the blood vessel. But whenever you have an injury like that, you're almost always injuring tissue cells as well. And then they're going to release the tissue factor that sets off that extrinsic pathway. All right, so when we have prothrombin activator, that's what we made. Once you have that, that's an enzyme. You know, we had to get here. And it takes prothrombin. Prothrombin is another one of these procoagulants that you have in your blood. It's also known as factor 2. That's why they're showing you the Roman numeral 2. This functions as an enzyme. And it converts prothrombin to thrombin. Okay, thrombin is also an enzyme. And what it does, once you have this, it does a couple of different things. It converts fibrinogen, which actually fibrinogen is about 4% of your blood proteins that are circulating around at any given time. You have lots of that available. And it's inactive, it's soluble. You see what they're calling it soluble here on the diagram. That means it's dissolved in your plasma, so it's not forming a meshwork. Thrombin converts fibrinogen to fibrin, and fibrin cannot stay dissolved in your plasma. It falls out of solution and forms a fibrin meshwork that traps red blood cells and platelets. So it's an insoluble polymer. Those fibrins get networked with each other. They form a polymer where you have lots of fibrin subunits that get linked together. Okay, thrombin also, in combination with calcium ions, so calcium, very important for blood clotting, takes yet another clotting factor called factor 13, activates it, and then factor 13 is what takes these fibrin proteins and cross-links them together so that they form a netting or a meshwork. So finally, we have made our blood clot. I do want you, though, to know phase 2 and phase 3. I do want you to know that story. And I also want you to know what's going on over here. That the intrinsic pathways and extrinsic pathways, know what sets these off. Know that both of them are leading to activated factor 10, and then all of these components that you see down in here, activated factor 10, calcium, platelet factor 3, activated factor 5, they all wind up getting together to make prothrombin activator enzymes. Prothrombin activator enzymes then set off the phase 2 that you see here. They convert prothrombin to thrombin, which then leads to these events happening and you make that blood clot. All right, so none of this would happen unless you start off at the very beginning with the extrinsic or intrinsic pathways. Okay, little trivia here. Notice once you go through the intrinsic and extrinsic pathways, or both, and you have prothrombin activator enzymes around, it takes about 10 to 15 seconds after that for a clot to form. And you guys know that if you've ever, when you cut yourself, if you're healthy and you have normal blood clotting, it forms pretty quickly. It's a pretty fast process, which is good for our survival. All right, and I think also even on this slide, I've already covered a lot of this. Your fibrin is insoluble, so it comes out of solution. It's no longer dissolved in your plasma. And here's another nice little image that was made with an electron microscope, which is showing you a bunch of red blood cells that have been colorized so you can see them better. And all this brown netting type stuff you see there is actually the fibrin proteins that have come out of the plasma and have formed that meshwork trapping those red blood cells. All right, so the next video lecture, once you form a clot, how do you get rid of it? And we also have to make sure that once you start forming a clot, that is a positive feedback process. You want to make sure your blood clot only forms in a local area. You don't want that blood clot growing and growing and growing and growing and becoming so massive that it winds up cutting off your blood flow, because that would wind up killing you rather than the bleeding to death, which was your initial problem. So on the last video lecture about coagulation, we're going to talk more about how blood clotting is kept under control and how you dissolve a clot once you're done with that clot.